Hello, I'm Mina Bissell. I work at Lawrence Berkeley National Laboratory. It is one of the first national laboratories in US, but is part of the UC campus, so we are a campus of University of California. I'm delighted to be giving this talk to you because, as you will see, a lot of what I'm going to tell you is still not in your textbooks. So I'm delighted to be able to talk to you and explain why I got so excited about it and why it's so important. So the title of the talk is The Importance of Extracellular Matrix and Microenvironment in Tissue Specificity and Breast Cancer. So the part one, which is more general for a more general and less experienced audience, is that half the secret of the cell is outside the cell. And so the talk is going to say that the genes and the microenvironment actually talk to each other. So to try and start this, I work in a national lab, and very often I have physicists in my talks. So I try to tell them what I do, and I give them the lesson one in developmental biology. And what is lesson one is when your mom and dad met, there is an egg and there is sperm on top, that little blip. A single cell gets fertilized. It does this. It becomes double, quadruple, moves on. It then becomes an embryo. And before you know it, look what it does. It makes this. So the question is, how do these cells that all started from that single one, all of them with the same genetic material, how do they make her the way she is? Of course, not the lipstick, but all the rest. Now, have a guess. How many cells do you think are in her body? So the opinions vary. Some people say 10 trillion. Some people say 70 trillion. I don't know who counted them, really. But the point is that it is more than the national debt of US. Now, every one of these cells have the same genetic material. So the question is not only how your nose becomes your nose and your finger, but how do they remember to be a nose and a finger? How do they do that? It's a very important question. So, and all these cells have to coordinate in a way to make you who you are and to tell them to remember what they do. Now, many years ago when I used to say that, people would say there is such a thing as terminal differentiation. Well, one morning you got up and what you saw in the front page of New York Times or your local paper was that somebody took a cell from a mammary gland. Now remember, it's the mammary gland. A single cell from a frozen mammary gland, and what did they make? A dolly. So it says that that particular cell has all the information to make a whole sheep. So why don't we get up one morning and have our nose turn into our elbow? It doesn't, right? Okay. So I came from a background of chemistry and bacterial genetics. And I really had not taken any biology during my career, some biochemistry, but not very much biology. So I came to do my postdoc working with what is known as Rous sarcoma virus. This was discovered by Rous in 1911, and I'm going to be referring to it as RSV. It is a chicken virus and forms ugly tumors like this in the chickens. I mean, look at it. Rous discovered this, and then he made the Koch's postulate to come true. In other words, what he did was he took the tumor, he made a filtrate, and then he injected the filtrate into the chicken, and when he did that, he got another tumor. So everybody got so excited about this, and when people defined what was the cancer gene, then they could even just inject the cancer gene and still get a tumor. So the whole wonderful, exciting field of oncogene and tumor suppressors got created because of this discovery and because of the later discoveries. But in all honesty, it didn't make any sense to me. We have 10 to 70 trillion cells in our body. The chicken has quite a few too. So you inject a single virus into the chicken wing and you get this huge tumor and you kill the chicken? This doesn't make sense. Every time you breathe, every time you go out in the sun, every time you get depressed, every time whatever, you get wounded, whatever, you have mutations. And some of these mutations are quite deleterious. So if every time you had some oncogene activated, you end up with a tumor, you would be tumor, tumor, tumor all over, and you're not. So what is going on, and why does this appears as if it's a single step, single oncogene? So I did an experiment when I had my own lab at Lawrence Berkeley lab. And what we did was that we injected this virus into the wing of a chicken embryo. So we could visualize the embryo inside the egg, and we could inject the virus in the embryo. And we did it for a reason that I don't have time to tell you, but you can read the original papers. And we showed that the virus didn't cause tumors in the embryo. And we showed that, in fact, the oncogene was active. We published this in a fancy journal like Nature, 1984, and not irreparable. Because, and I don't blame people, the burden of proof was on us to go and actually show this. But as you see here, we took the oncogene, we made it blue by attaching it to a reporter gene. And then in order to make people to really believe that oncogene is active, every one of these beautiful cells, blue cells in that wing of the chicken, in that feather, could in fact be a tumor, and they are not. Now, if we dissociate the wing and we put it in a dish, we get mass transformation overnight. So what on earth is going on? In the chicken, you get a tumor. In the embryo, you don't. You put it in a dish and you get mass transformation. What does that tell you? And as you see, we published this in many different journals, and they were well-known journals. But again, nobody would pay attention. But this told me something very important, and that was that the context, i.e. the microenvironment, determines what even a potent oncogene can do. But what is context made of? And does it signal? And besides, people really were not interested in chicken and chicken viruses. They wanted to really talk about large genes, and they wanted to talk about mammals. So I decided that I'm going to really be spending a lot of my time and life answering the following questions. How is tissue specificity maintained? I.e. noseness, fingerness, elbowness, prostateness, mammaryness. How is it maintained? How does one study the problem in mammals? Oh, the number of times I wished Drosophila would have mammary gland so one could do genetics, but they don't. And how is the program lost in cancer and aging? It is precisely when a cell doesn't know that is supposed to be a nose or a finger or a mammary gland that it starts to pile up and go elsewhere. And aging is the most important risk factor in cancer. And these things are related, and they have something to do with the title of my talk, which is the microenvironment. And finally, of course, how can one use the information for therapy? I'm not going to be able to answer all these questions in my introductory lecture, but I will answer some more of these in my next lecture. So to model mammals, we turn to using the mammary gland as an experimental organism. It is one of the few tissues that changes during the adult life of the organism, and it moves again and again, and it is an organism within an organism. How? Because it doesn't develop in the embryo. It only happens when the young women start getting their puberty hormones. So both men and women start with the onlaga of the mammary onlaga. Then the hormones come, and the mammary gland grows and grows and grows and grows, and it's puberty with those little pretty things at the end. Then the animal starts getting pregnant, and then it buds and buds and buds, just like the clusters of grape. And then when the animal is pregnant, it looks like that. And when the babies are taken away, after they suckle, the whole gland goes back to this and travels all the way around. So it's a magnificent model system to answer this question that I have raised for you. So we decided that the mammary gland itself is quite complex. Here on the left, here, you have this one single lobe of the human mammary gland, but the mammary gland, as you see, has a nipple, has duct, has branches, has all of these other stuff. And then when the animal gets pregnant, at the tip of that thing, of the branch, there is what is referred to as an acinus here. It's a very lovely organization, has a lovely organization. It has a basement membrane which is made of extracellular matrix molecules, large molecules that are outside the cell, like collagen, like laminin, all these things that keep you together. If you took the collagen out of your body, you're going to be like Jell-O. So people always believed these things had no information and they were just structural elements. Now, you have that lovely basement membrane around. Then you have one layer of epithelial cells that are beautiful and polar, and they secrete the milk inside. But between the basement membrane and the epithelial cells, the luminal epithelial cells, there are those blue cells that are called myoepithelial cells. And these cells squeeze the gland, and when they squeeze, the milk comes out. Okay, so we said, wonderful, we are going to model this, and we are going to say, how does the mammary gland do it? Now, no sooner we had done this, that we looked at a single cell of the mammary gland, and we could see that it was beautiful and polar. It had secretory activity. It had a beautiful basement membrane. It had a nicely organized nucleus right there. And it was secreting gobs and gobs of milk. So we take these cells, we put them in a dish, we give them all the lactogenic hormones, we give them everything they need, growth factors, whatever, no serum, trying to keep them functional. In no time at all, they look like this. As you see on the right, the cells lose shape, they lose polarity, and they completely forget to make milk. And you say, you silly cells, 2 days ago you were in the mouse, you were making all this milk, what happened to you? So it says that the function is not permanent. If you change the context, the function changes, right? So we said, what is missing? So we made a section of the mammary gland, and as you see, here's a section of the mouse mammary gland. There are all those beautiful acini surrounded by the red, which is marking the basement membrane, in this case laminin, laminin 111, a very important extracellular matrix molecule that is part of the basement membrane. And you see that the center is where the milk is, and you see these big globule things in here that have fat in them. So I began to think, and this was late 70s, early 80s, maybe this material has information, and maybe it's telling the cells what to do. And just around those times, we didn't even know that extracellular matrix molecules either had receptors or they could actually signal. So we said maybe this material is important, and to make a very long story short, we made a gelatinous basement membrane that we isolated from a tumor. And there is a woman called Hinda Kleinman who had done initial purification at NIH. And we took these tumors that we used to make in my lab, and now they can sell it under the name Matrigel, and they can make a lot of money. They have made a lot of money, and I wish we could have patented it. We would have made a lot of money. But we didn't. But nevertheless, we put this stuff on a dish. We put the cells on top of it. And as you see, in no time at all, they go from this clump, they pull this material over them, they reorganize, and they make that beautiful acini. This is in culture, and this thing is actually in vivo. And even the sizes are similar. So we then said, is it only looking lovely? Is it also looking polar? It puts the milk inside and it puts the other stuff outside. Does it actually look beautiful like this, as you see, how gorgeous they look? Or do they also make milk? They make gobs and gobs of milk. Now, on the left is what they make on plastic. On the right, the red is all the milk that is inside, and the blue are other things that go outside. We actually labeled this and did this experiment. Okay, now if you look inside these beautiful acini that have come together, you see that it's full of milk. Now, I am getting up there because I told you I was doing this experiment in 70s, etc. And people in my lab did a wonderful birthday celebration for me for one of those big birthdays, and they did this. Now, for those of you who don't read the American newspapers and don't know the ad for milk, this makes the American audiences crack up. But I have a hard time doing this in Europe because they don't understand it. But anyway, so we got milk, and we have a lot of milk. Now, given that normal cells can remember how to look in 3 dimensions, and they can make milk, and they remember that they're mammary gland, we said, could tissue structure also be a message for the tumor cells? We didn't have any assays for distinguishing normal and malignant cells from each other, okay? So I thought, gee, maybe we could do this. And I did this for humans to also have a counterpart to the mouse after a wonderful guy called Ole Petersen, who is now a Her professor in Denmark and the head of the department, etc. He came to my lab and he said, I really would like to do a sabbatical in your lab. I want to work with you. He was an MD PhD, so I said, okay, let's take cells from reduction mammoplasty. This is when women have large breasts and they do it to themselves. They go and take a piece of it out, and usually they used to throw it in the garbage. So we could take this material, we could separate the cells and the material, and we said, can they actually make the same kind of the organization? And they do, and they look gorgeous. And then, as you see in this movie, on the top, they are the normal cells, and in the bottom, they are the malignant cells. And Ole did this with primary cells as well as with cell lines, and we could show that every one of them. Now, the ones on the top, remember, they are not actually single cells. They are like that lovely acini on the black top on the top. But the ones on the bottom are the tumors, and every one of them make an ugly tumor. So it's like having hundreds and thousands of transgenic normal breast acinas and thousands of tumors out there, and they are clonal. So it's a wonderful way of being able to take them out, do all sorts of things with it. And if I would have had time for you, I would have told you a fantastic screen we came up doing just using this concept, which is now has been sent out for review, but is not yet published. Okay, but you know, I did not want to be doing these experiments comparing a normal cell to a malignant cells where they came from 2 different individuals. They came 50 years ago. They came at all those, and there was no comparison. So we took advantage of a very interesting experiment that Ole Petersen and his professor had done. They had taken cells from reduction mammoplasty, as you see from this woman right there. Then they put these cells in a dish, complete defined medium, and they grew it and grew it and grew it only in the presence of epidermal growth factor. Now, at some time around this time, they took the epidermal growth factor away, and I kind of came into the picture at that time, and they started injecting the cells that were grown in the absence of the growth factor, and they started injecting it into the animals, and they eventually got a tumor, and the tumor could go back. So what do we have? We have the first model of human breast outside humans. We have every step of a cell going from normal to malignant. And we have so many passages, and we have them frozen, so it's a wonderful progression model to tell us how do they do it. Now, the non-malignant cells, as you see, are called S1. The pre-malignant cells are called S2. The malignant cells are called T42. And when I was looking at these under microscope, they all look the same on 2D, on plastic. Now, if I take this progression and put them in 3D, that's what they do. So I'm going to take you through this, all right? So let's start with passage 50, which is the passage that we actually use in culture. As you see, they already have amplification and deletion. I did these in collaboration with people who do comparative genomic hybridization, and so I did it with Ole Peterson. Now on passage 110, as you see, MYC amplifies on passage 50. In passage 50 already, P53 got lost. All sorts of things are happening, but the cells still say, hey, I'm normal. I can make this beautiful structure. Even in passage 175, they get a little bit bigger, and they have a whole lot of other mutation, but they still think they're normal. Now, the exciting thing are the pre-malignant cells. We all of a sudden have an assay for pre-malignancy. As you see, those are called S2, and they already lost the structure. That's very interesting. It is before malignancy. The structure is going before malignancy. And as you see, they have a lot of mutation, but they're not yet malignant. Once in a blue moon, we got that tumor that I showed you, right? The one at the end. And that one is also a mess, but something happens between S2 and T4 that makes them malignant, but the structure goes first. Now, if you think about it, when I destroyed the structure of that chicken embryo wing, they became malignant. When I had it in the beautiful feather and the beautiful wing, they thought they were normal. So here is similar ideas. So we then said, and we made this very crazy hypothesis. We said, if tissue structure is the message, we can hypothesize that tumor cells, even with abnormal genome, should be capable of becoming phenotypically normal if the structure is restored. And then the destruction of tissue structure by itself could be a carcinogenic event. Now, this was crazy because I was saying we could make a malignant cells by making them remember their structure, making them think they are normal. Alternatively, we could take a perfectly normal mouse, mess up the structure of their mammary gland without oncogene, without suppressor gene, and make them malignant. Well, we have done both of these, and I'll now show you a little bit of data on this. So on the left are these beautiful acini of the non-malignant or normal cell. The reason normal is in quotation is because we use cell lines from time to time, but everything I'm telling you has also been done with primary cells from human breast from reduction mammoplasty. And on the right, you see these ugly cells that were derived from there. They have something in common, you mean the ancestry in common, but one is completely messed up. Okay, we said, how does the normal know and the tumor doesn't? How does that happen? So they get the message from the microenvironment, right? So Valerie Weaver, who was a postdoc in my lab and now had her own mini empire at UCSF, she basically started taking these cells and measuring what was on their surface. Because it's the surface that is allowing things to go in and out and messages be taken from the extracellular matrix and microenvironment. So we showed that the tumor cells and normal cells have a lot in common. They have the same kind of integrins, they have the same kind of the epidermal growth factor receptor, but the ratios are different. The malignant cells had something like 6 times the level of beta-1 integrin, 5 times the level of EGFR, et cetera, et cetera, et cetera. So for various reasons, we decided to go back and make the malignant cells have the level of beta-1 integrin on their surface, which is equivalent to the level in the normal. okay? Not to 0, but equivalent to the normal. And we used an inhibitory antibody against beta 1. When we did this, look what happened. The cells stopped growing, they reorganized, they're not 100% normal because they have all those mutations, right? And to just show you that we didn't select, here they are, every one of those on the top malignant, every one of them on the bottom reverted. We call this reversion. Now, if we take these cells on the bottom and inject them like that into the mice, 0 tumors. There are 100% tumors if we do these cells on the top, right? So, I would show this to people and people would still say, no, you must have selected, you must have selected. What selection means? How did I select 10 to the 7 cells or 10 to the 6 cells in 3 days? So we had to again do the darn experiment, another experiment to make people to listen to us. So we did this. We said reversion is reversible. What did we do? We took cells that were taken from the monolayer, and they, on the top, we put them without antibody. You see that ugly picture. And in the bottom, you see that we reverted every one of them. Okay. We then took these cells, dissociated them, put them in another monolayer, and the one without the antibody, again did that on top, and the one with the antibody did this on the bottom. So we can do it again and again and again, showing that this is not a selection, and the conclusion of these studies is profound. Is again what I told you in the beginning, and that is that phenotype is dominant over genotype. Now, this used to drive people up the wall, but I would say to them, look at yourself in a mirror. The same gene, the same gene, the same gene, the same gene. So why couldn't tumor cells have the same gene and entirely different shape, which is exactly the same thing? All right. That being the case, our conclusion was that growth and malignant behavior are regulated at the level of tissue organization, and that tissue organization is dependent on extracellular matrix. And I wrote a theoretical paper long before this in Journal of Theoretical Biology in 1982, and I suggested that the outside talks to the nucleus and chromatin. Chromatin reorganizes, talks through the cytoskeleton to the outside, and the whole thing is dynamic and reciprocal. So, more than 200 publication reviews from our lab in different aspects of this statement I just gave you, and please Google us if you're still skeptical. So, one other thing to tell you. Signaling in 2-dimension and 3-dimension are entirely different. And I'm just showing you one very interesting example. Now remember, until very recently, the drug companies are trying to find cancer drugs and they test them on 2-dimension. And we show that the top blot over there is Coxsackie adenovirus receptor. And as you see on the bottom, the S1 and T4, one non-malignant, one malignant, in 2D, they're identical. Alpha V integrin on the bottom is identical. Now you take the S1 and you put it in the 3D and look at it. It's like day and night. It just goes down, and then the tumor cells still remain high, and then when we revert them, which is the last thing on that side, you see that it is reverted and is in 3D, and so it says that the cells are completely dependent on the context and they are plastic. Because if you look on the bottom, alpha V integrin, which is needed for the activity of Coxsackie adenovirus receptor, is the same in 2D in normal and malignant cells. But when you put them in 3D, even the tumor cells now upregulated. Now this is very exciting, and it says that regulation in 2-dimension and 3-dimension are not the same, and nothing about you, absolutely nothing about you in your body is two-dimensional, right? So that's why they're able to do the kind of things they do due to the shapes that they have. And I come back to that later in my next talk. So there was something very interesting happening, because what was happening was that we were actually giving the cells beta-1 inhibitory antibody. Now, these cells had 6 times the level of EGFR, 6 times the level of EGFR. How did they stop growing? What happened to that? So we measured EGFR, EGFR was down. So we then use inhibitors of EGFR that make beta-1 go down. So it said that these things are talking to each other, and when we looked at the same phenomenon in 2 dimensions, they weren't doing it. Now we have done the experiment with many, many different inhibitors. As you see, beta-1 integrin is making the tumor cells go down and revert. You see that EGFR does the same thing. You see that MAP kinase does the same thing. You see that PI3 kinase does the same thing. If I go off the screen, you can see it. And you see that if you put back dystroglycan, it does the same thing, notch, taste, etc., etc. What then is an oncogene? All these pathways have to integrate. And many of these pathways have oncogenic activity if the balance of the signaling becomes awry, it goes awry, right? So we can also take metastatic cells and we can inhibit them with 2 different inhibitors. That being the case, I would argue that if you gave me any of these breast cancer cells, I'm going to be able to put it in this assay, understand what was outside on the surface, and I could revert them. All right. So form and function are related dynamically and reciprocally. You mess up one, you mess up the other. This is really important to think about, both at the level, it's not just your mind and body. It has to do with every atom and every single cell of your body, that the form and function have to be integrated. What gives the signal to the mammary gland in vivo to tell it to be polar and functional? And what substitutes for laminin-rich gels in vivo? Now, if you look at the structure of a mammary gland, as you see, I love this picture, it's a section from a mouse mammary gland. You see one of those beautiful acini in the middle, and you see that there are luminal and myoepithelial cells, and then around it is a basement membrane, and all those white stuff are the fat in the mammary gland. So what surrounds the cells that make the milk are the myoepithelial cells. So together with Ole Peterson, we began to think maybe it is myoepithelial cells that gives the structural cues to make that lovely structure. They don't have matrigel in the mammary gland. So you see in situ, if you look at it, you see that that beautiful picture of one layer of epithelial cells, one layer of myoepithelial cells around them, and they are actually shown, they are highlighted in red. Now, if we dissociate and separate the myoepithelial cells and the epithelial cells using magnetic beads and purify them, before we can do that and we put them on the dish in culture, you see that the myoepithelial cells start looking like a muscle, and the epithelial cells are behind. And so we then separate them, and then we do a classical experiment. Now, we can't do the experiment and test the action of myoepithelial cells in actually laminin-rich basement membrane, because as you have seen, in laminin-rich basement membrane, you already get this. So we said, because laminin-rich basement membrane gives signal, right? So we put them in collagen 1, which if you only looked at them, you see that they look the same. But if you section through these 2 very similarly looking structures, on the bottom you see that this one has a lumen in the middle, while that one doesn't. The one on the right doesn't. Okay, so we take the luminal epithelial cells and put them in the basement membrane gel, as I have shown you. On the top, you see luminal epithelial cells, we are calling it LEP, in the basement membrane. They have epithelial-specific antigen shown in green, and then sialomucin shown in red on top. And on the bottom, you see the nuclei in red, and then you see that beautiful basement membrane in green. Now, you put the same cells in collagen, for crying out loud, look at it. The cells in collagen are inside out. Even though they look the same, even though they both had to stop growing, they are not getting the right signal from ECM. Collagen is not laminin, and ECM is not ECM. They give different signals at different parts of your body. And so these same cells in collagen are doing that funny thing. On the right, as you see on the right, you see that they are pouring out tremendous amount of basement membrane molecule. And when they do that, you think they should make a basement membrane. They don't, because they're inside out. They don't have the right structure. So now we can test the function of the myoepithelial cells if we add it to the collagen. We can't add it again, remember, to the basement membrane because they already have that pretty structure. We want to know what gives the signal in vivo, right? So we now add myoepithelial cells and look at that. On the left is in collagen without myoepithelial cells. On the right, we have the epithelial cells, and they're gorgeous. They make the double layer of the mammary gland, and now they make a lovely basement membrane in green on the bottom. The one on the bottom is called the double layer tube. Okay, we published this, it was very exciting, but then we wanted to know how did myoepithelial cells do this? So when we looked at that, we realized that LAMO-A1 chain, and now I'm going to be giving a little bit more science-specific detail, just for those of you who are just dying to see gels. So here it is. So here are the luminal epithelial cells in basement membrane on the top. They have no LAMO1, laminin A1 change. While they have the LAMO3 and LAMO5 and collagen 4A1 and even collagen 4A2, although it's a little bit low in normal. And then the LEP in collagen, they have all of those. Again, they don't have LAMO1. But the myoepithelial cells are the only one that have that lovely band on top. So all of a sudden, everything came together for me. How exciting. Myoepithelial cells in vivo make laminin. And that's why in the laminin-rich gel, we are able to substitute for myoepithelial cells without having the myoepithelial cells. So this was quite exciting. And then we could show that in place of myoepithelial cells, we could add laminin 1, and when we did that, again, they made these beautiful organizations that I showed you in these acini, and if you use all other kind of laminins or type 4 collagen or fibronectin or whatever, you won't get it. Now, the exciting thing came when we look at tumor cell myoepithelial cells. We realized that the tumor cells, 70% of them, were not making the laminin A1 chain. They weren't making laminin. And some of them were making a little bit, and that's shown here, but you can read it in the paper. It came out in Journal of Cell Science in 2002 from Ole Peterson and my laboratory. And as you see here now, on the left is one of the tumor cell myoepithelial cells that could partially revert. And on the right, you see that they look as if no myoepithelial cells was added. They look just like plain luminal cells inside. 70% of tumors cannot do this. It's quite important. And then we went to the human patients. On the top, you see the normal, beautiful acini with laminin around them. And then you take the other 5 or sections of the tumor, laminin basically gone. So that's one of the reasons that the tumors don't do well. They're not getting the right message. And remember, in breast cancer, one of the things that happens is that myoepithelial cells get lost, and nobody still knows why. And it probably has a lot to do with the stem cells. It has a lot to do with others. But more of that later, and hopefully somebody else comes and lectures to you, although we also have some data at that level. Okay, so let's see. Now, does this have what we found in culture? Is that reproducible in vivo? And very well-known and talented scientist at Harvard, a good colleague of mine, Cornelio Polyak, had a paper in Cancer Cell in 2008, and I'm one of the 17 authors, but it's really from her lab. And what she did, she made a model of ductal carcinoma in situ. And what she did, she put luminal cells together with either fibroblast or with nothing, or with tumor myoepithelial cells, and then injected them into the mouse, and they always became malignant, and they became invasive ductal carcinoma, even if she gave them normal fibroblast. But when we sent her some of our normal myoepithelial cells, the only thing that could prevent that arrow from going to the left or to the right, in this case, depending on how you're looking at it, is normal myoepithelial cells. So that was very, very gratifying. So finally, does loss or aberration of ECM have a functional significance? And if so, can it lead to tumor formation? And so we did experiments with Zina Werb, who is a professor at UCSF. We made a mouse in my lab where we made a conditional mouse where we took a milk protein promoter, attached it with a metalloprotease that destroys the basement membrane and ECM called MMP3 that was occurring during involution. And we showed that we could indeed make almost a conditional mouse that when it got pregnant, because it had the milk protein promoter, it could now upregulate MMP3 in the middle of pregnancy, and these mice, as they aged, got tumors. Look at it. They have tumors. Now remember, no oncogene. No, at least not in the beginning. It doesn't mean that later they don't get it, but at least at the moment, we started simply by messing up the structure. This was quite exciting. We published a lot of these data in Cell in 1999, and tomorrow I'm going to talk a little bit about the mechanism that we uncovered and a lot more detailed about how this happens. So we have done both sides of that equation that I started from. You restore structure, the cells think they're normal. You take a normal cell, destroy the structure, they become malignant. This can explain actually what happens when you have asbestos, because like a chronic wound, it's constantly changing the structure. When people have inflammation in their body, they're constantly getting the wrong signals, and we are now learning that a lot of these things are important in cancer. I was in France doing a sabbatical, and it was Jacob and Monod were two Nobel Prize winners. And these Nobel Prize winners, one of them, Jacob, or was it Monod, said, what is true for E. coli, and E. coli is this little bacteria, is also true for elephant. Meaning that if we study E. coli, we'll get all the answers for the elephant. That is actually true 80% of the time, however, is not true all the time. And this cartoon that somebody drew for me shows E. coli on the top and the elephant on the bottom, and it gave me great pleasure to show it to the French audiences. I would say, the E. coli is saying, is it really true that what's true for me is also true for the elephant? And what I like to say is that what is true for E. coli may be true for elephant, but what's true for elephant is not true for E. coli, because when two higher organism cells come together, the result are logarithmically different. The cells come together, form junction, have a bottom, have a top, have secretory activity, have different structure in their nuclei, have a different dynamic reciprocity. So we have to be cognizant of this as we keep treating our cells in culture as if they are E. coli. They are not. The question is, how do we now model metastases? A lot of people are using three-dimensional models. Even companies are using three-dimensional models. They are using it for testing drugs, which is wonderful. And we thought that maybe we want to make a model for metastases. And we again want to use this invasion of the mammary gland into the fat pad as a model. And that I will discuss at the end of my next segment. So, and that's shown here, the mammary gland whole mount on the top and the higher magnification on the bottom. And I want to finish with this very favorite cartoon of mine where the guy is looking in his very, very formal suit and telling the cat, never, ever think outside the box. Now, I used to be the cat, and those were the authorities. Sadly, I have now become an authority. So I don't want you to listen to me either, but to always, always think outside the box. And now I will just make some general comments about cancer. The reason this is so important is because even though we know a lot, we don't know enough. There is so much we don't know that is not even funny. And here, if you look at this article that came out by Clifton Leaf in Fortune magazine in 2004, and I hate to tell you that even the current data haven't changed that much. We are making a lot of progress on some aspect of breast cancer, but many different kinds of cancer we are not. Heart disease has gone down between 1950, and now at that time, quite a bit, and cancer mortality. I mean, death from heart disease has gone down. Cancer mortality, if anything, has remained completely steady. So we have a lot to do. And this is why I encourage all the students and others to do a lot more. I usually dedicate my talk to this gorgeous woman who was a friend of mine. She modeled this dress few months after her breasts were removed, and she struggled wonderfully, but it was too late for Sonia. I do not want it to be too late for my granddaughter. Here she is, Chloe, when she was 5, and she's now 11. She always says, Grandma, why do you keep showing the same picture? And in all honesty, I haven't had time to change it, even now, even for this. So here is Lawrence Berkeley Lab, and I usually, when I lecture, you know, we have Lawrence Livermore, Lawrence Berkeley. This is, and even the secretaries of energy sometimes mix up Lawrence Berkeley Lab and Lawrence Livermore. It drives me absolutely crazy. Anyway, so here is Lawrence Berkeley Lab. We are part of the University of California. We have that beautiful Golden Gate Bridge. We don't do defense-related stuff. Not that I have anything against doing defense, but we don't do it. We do multiple things, including biology, as you have gathered. And when Lawrence, again, I said it's the first national lab in US. When Lawrence smashed an atom, he was using a building that had the accelerator on the bottom. Then they kept the dome and they put it on the top of a very intense synchrotron radiation beam called advanced light source. And those of you who do structural biology, come see us. And so that you will never forget and not mix up Lawrence Berkeley Lab and Lawrence Livermore. What does this remind you of? Does it look like a breast? Now, only women get away with this, and this is for you to never, when you come to Berkeley and you look up, you will say, oh, that's Lawrence Berkeley Lab. And here is my lab a few years ago, actually. But that tomorrow I will talk about some other work of each one of these kids. But the general picture that I have given you has been done because of wonderful students and postdocs over the years. And I love many of them, in fact, most of them, almost all of them. They're all over the world. And again, tomorrow I will talk specifically, or I keep calling it tomorrow, but in my next lecture, I'm going to be talking about a specific work of some of them. And I spent 3 months in France recently, and I came back, and as you see, we actually take the mammary gland seriously. 8 of my fellows, either the guys with their wives or the wives who were my own fellows, had a baby, and they made me a human as an eye. And where do we go now? And that is part 2. Thank you very much for your attention.